here. Of course, we have no chance with big data, but we will also have some, let's say, a little bit of big data and some of the history. So let me start. Maybe the wrong one. Yep. Oh, sorry. Okay, so I'll start with some history. And of course, you know the Philadelphia chromosome story, but I would like to stress here that actually leukemia was first in many aspects of oncology research and not only oncology research. And Mel Greaves has, let's say, published an excellent review in Nature Reviews in Cancer 2 years ago. And as you notice here, the first chemotherapy in acute lymphoblastic leukemia, the first discovered oncogene, the first targeted therapy, the first stem cells discovered in human cancers, all those is related to leukemia research. And it is also very, very abundant presence of the very rare and orphan disease, chronic myeloid leukemia. And I would like to start with this disease, which I'm studying for almost 20 years. Philadelphia chromosome story, I'm sure most of you know very, very well, and the novel and hunger for discovery of the Philadelphia chromosome when they worked in UPenn. But I would like to go back a little bit, and actually we will go back to the 19th century, and two gentlemen, John Bennett and Rudolf Virchow, who almost at the same time discovered most probably the first human leukemia patients. They are first human leukemia patients published. As you notice, they published this almost in the same time. Today, probably one of them would be suspected of plagiarism, but at that time there was no PubMed, no internet, so I'm sure they published this independently. Leukocytemia, that was the title of John Bennett's work. Weißes Blut, which means white blood, is the title from Rudolf Virchow. Why? Because we suspect it had to be chronic myeloid leukemia, because actually this is the only leukemia which at the diagnosis can present with 50, 60 times more leukocytes than normally. So instead of 10,000 leukocytes per cubic milliliter, patient may be, let's say, maybe diagnosed with 500,000 or even more leukocytes, which make a strange picture that the blood is not really red, the blood is kind of white. So that's why we think they both had patients with chronic myeloid leukemia. Later the story goes on, and then we have a young doctor. Yes, this is the same doctor who wrote the Sherlock Holmes. He was an MD, and he also described a very interesting case of leukemia, which was most probably chronic myeloid leukemia, and the high doses of arsenic at diet was very effective. So the causative link was actually wrong, but more than 100 years later, actually arsenic trioxide became a standard of care in a rare subtype of acute leukemia called acute promyelocytic leukemia. Arsenic trioxide induces differentiation of this leukemia. Let's come back for a moment to Novel and Hungerford. Of course, as you know, when they discovered this small chromosome called myeloid chromosome in the 60s, they had no tools. So we had to wait till Janet Rowley went for her sabbatical to Oxford with her two young daughters. And then as she was, let's say, telling this story at one of the last ASH meetings she attended when she was more than 80, she was coming back very tired home from the lab, and her children were waiting for her, her two daughters. So she was trying to figure out what's going on with those chromosomes, making jigsaws with her daughters. And this is how she got the idea of those two famous translocations, the AML-type in acute myeloid leukemia and also Philadelphia chromosome. She published this in 1973. First acute myeloid leukemia, famous translocation, then Philadelphia chromosome. So we know very well now the pathogenesis of the disease, and we know this is also the first disease when we have a targeted therapy. This gentleman in 1998 was the first patient, the operator of the train from Oregon. He received the experimental compound STI-571, which later on became the famous imatinib, the first targeted therapy. Actually, it's not the first targeted therapy, but it was the first kinase inhibitor, because we had different other kind of targeted therapies before, like monoclonal antibodies. But here I have some remark that always there is some perseverance behind any this type of success. And I would like to acknowledge John Goldman, who was in the 90s the chief of the bone marrow transplantation unit in London. He was the head of hematology. He was very influential. And when he got this first initial data from Ciba-Geigy at that time, it was not Novartis yet, Novartis was, let's say, ready to close this. They didn't want to open the clinical trial because at the beginning of 90s, nobody believed that tyrosine kinase inhibitors might be effective in human cancers because everyone thought they will be too toxic. So he personally went to Basel and convinced the chiefs of Ciba-Geigy at that time pursue the clinical trial. And they said, okay, we will start it, but you know, in Europe it will be very complicated. We will start it in the US. So you will not be the principal investigator. And he said, okay, but go on with it. And this is how Brian Drucker became the first author of the famous IRIS trial and the New England paper. And John Goldman, with, let's say, credited him this. So this is one of the amazing stories, and he was one of the mentor for hundreds of hematologists. So now some questions which I would like to raise, because for many of you, this disease seems to be, let's say, the solved case. Okay, we have the imatinib, we have several other tyrosine kinase inhibitors, so why do we need to study this disease anymore? Especially if you look at the, let's say, one of the biggest clinical German study group. So now with the tyrosine kinase inhibitors, we have people, more than 90% of the people who are living more than 10 years taking one pill a day orally. So we would love to have such situation in several other cancers. But we still have few patients who will progress despite the therapy with tyrosine kinase inhibitors to the so-called blastic phase or blast crisis, which resembles acute myeloid leukemia and which is unfortunately fatal. So usually we don't have time when patient progress to blast crisis for allogeneic stem cell transplantation. So this is one of the reasons we still have few patients which have, let's say, this risk of progression to blastic phase, and I'm going to come back to this issue later on. But also we have another goal in this disease. We would like not to treat patients forever, we would like to cure them. And actually this is, again, a prototypic disease when such attempts are now performed for the last 8 years. French guys started in late 2008, 2009 with the famous STEAM trial, François Mahon. And now we have this kind of picture. In order to patient to be eligible to start such a trial of treatment-free, so we can stop the therapy, the patient must be usually at least 2 years in a deep molecular response. And so, which makes approximately 20 to 40% of patients eligible. And after some times, half of the patient will have to come back to the drug. So we don't know why, because of course all the patients enters this treatment-free period with the same criteria. So this is a very interesting story. Why half of the patients will relapse? Fortunately for them, they can come back on the drug. So there are only few cases described that, let's say, they stopped the drug, then they had the relapse, and they progressed to acute phase. Only few cases. Now, more than 1,000 patients worldwide has stopped the therapy, which is a big success. So we think that the problem is the number of the chronic myeloid leukemia stem cells, which are in the patient's bone marrow when they stop to take the drug. And again, I would like to remind you that acute myeloid leukemia, so again leukemia, not the chronic myeloid leukemia, was the first human disease where the real existence of the cancer stem cells has been proven by John Dick's lab more than 20 years ago. So in CML, we have a question, do we have to kill all those CML stem cells in order to cure the patients? So in this famous comparison to the Dandelion phenomenon, do we have to get rid of the roots in order to cure the patients? So here I would like to now show you some of the published research in which I participated, or our work. We are collaborating for many years. I'm collaborating with my former PI from Philadelphia, with Tomek Skórski, And in recent years, we have shown that actually CML stem cells accumulate more DNA damage due to the higher level of ROS production. And this is generated in mitochondria by the respiratory chain 3. And we have published this in blood, and we have also shown that even in patients who are on the treatment with imatinib, those CML stem cells which are resistant to imatinib. This is the major problem with CML stem cells, but even if the patient is treated with imatinib, they rely on different pathways, and they express BCR-ABL, but they are not, let's say, dependent on BCR-ABL. So in other words, the patient has a kind of a ticking bomb. And of course, we can argue, do we have to kill all the CML stem cells or not? But if we want to cure the patients, not just to treat them forever, we think we should rather kill all the CML stem cells. So there are several different pathways described in CML stem cells, and there are several different trials in the last 10 years which are attempting to kill the CML stem cells. They are not specific only for this type leukemia. They are also, let's say, similar trials are performed in acute myeloid leukemia, which is, of course, much more serious problem because this is, let's say, still a disease without a targeted therapy. But it was well summarized by Michael Deininger, so I think there's still a place for a lot of research in order to target CML stem cells. And if you think that it's not possible to publish, let's say, about CML stem cells and target therapy in Nature, it is possible. We are not so good, but in recent years, we have published, let's say, interesting story about one of the most often prescribed drugs for humans, which are statins used to treat hypercholesterolemia. And we showed that statins, by influencing the pumps in the membrane, they can keep higher concentration of imatinib in the CML cells. So in other words, simple method, and of course, if 20 million people worldwide are taking statins, and the median age for CML is 55, it's not very rare that we have a patient who is prescribed with statins and then develops chronic myeloid leukemia. And to, let's say, we have published this in 2014, and later on, we were very happy to see a clinical data from a team from Canada when they showed that people who are on statins, because of the hypercholesterolemia, are achieving better response, because here the probability of major molecular response, the curve means that those patients, let's say, have more chance to go into this treatment-free remission. So in other words, this is a rationale to use, let's say, well-known drugs in another scenario. But after Dr. Chowdhury's excellent lecture, I don't think I'm going to go here into details, but I would like to show you that we are also pursuing this concept of synthetic lethality in order to kill the CML stem cells, and not only CML stem cells. I like this comparison. Let's say the tumor, instead of 2 hands, has just 1 hand to eat, and we try to cut this 1 hand which has a tumor cell. So this is, I think, maybe the very simplistic, but I think very good example, I mean, good comparison. What is the synthetic lethality? And I will, if you allow me, I will just go very rapidly through the slides because after the previous lecture, you are well aware of those trials. What I would like to add to Dr. Chowdhury's excellent talk, that it's not only the ovarian and the breast cancer, but what he has just mentioned, we can now find such defects in, for example, pancreatic cancer. And we are looking for mutations in BRCA1, BRCA2, and other DNA repair genes. Only 5 to 10% of pancreatic tumor-bearing patients have this type of defect. But if they have this defect, they respond excellently to platinum-based therapy and also to olaparib. And with my fellow oncologist, we see patients whose, let's say, the diagnosis was very, very grim, and the expected median survival for them was less than 6 months. Some of them live more than 2 years on the platinum-based therapy plus olaparib. So our attempt was to use this concept of synthetic lethality by looking into such defects in leukemia stem cells, different leukemia. From our previous work, we knew that CML stem cells accumulate more double-strand breaks, and they repair them, but they repair them not really in the so-called faithful way. They rather use non-homologous adjoining on the so-called hyperactivated homologous recombination, which is not like a perfect way to repair the damage. So to make the long story short, we went on a fishing expedition and we tried to find these rare defects in different types of leukemia by the so-called GEMA attempt, gene expression and mutation analysis. And with this, we ended up after several years with last year with Journal of Clinical Investigation paper, which I was one of the collaborators. And this year, the follow-up was that in acute myeloid leukemia with flat-free internal TALNEM duplications, we can also propose this synthetic lethality concept to PARP inhibitors. So this is far too early to say if this will be the new treatment for acute myeloid leukemia, but we will try. Now some unpublished results. So of course, if you would be interested in NGS, you would be on the other room. So I'm not going to go into details of NGS. I would like just to tell you that we are using NGS. We rely on targeted sequencing, so we don't do whole exome sequencing or whole genome sequencing. We are not so rich, and we try to find what is the underlying reason. But I showed you before that 2 to 5% of the patients, despite tyrosine kinase inhibitors treatment, are progressing to blastic phase. And in our small cohort of 11 patients who progressed despite tyrosine kinase inhibitors, we found a very, very, let's say, intriguing observation. Most of them had pre-existing mutations. The inner cycle is the diagnosis. The outer cycle is the progression to the acute phase. So most of them had pre-existing clone harboring the so-called acute myeloid leukemia type mutation. Those of you who are interested in acute myeloid leukemia, RANKS1 transcription factor, isocitrate dehydrogenase, DNMT3A, those are typical mutations for acute myeloid leukemia. So in other words, we think that very small number of patients may be unfortunate to harbor such a clone. And now we know from the work of others, but actually some somatic mutations like DNMT3A may precede leukemia. And we also know from excellent Jaisval paper that actually we all accumulate these pre-leukemic mutations with age. And this is the question that if we will get the second hit in hematopoietic stem cell, then we may have a problem. And this was excellently described by Maggetti's group about the leukemia evolution, when we have the DNMT3A, usually is a pre-leukemic mutation, and then we got the second, the third hit, usually flat-free mutation or IDH, and then we go to the frank leukemia. So now, for the end, just a bonus track that sometimes we may be able to help right now and right here. We got a patient, and just one story of one patient. A professor from Wrocław sent us this case, a 60-year-old man who was suspected with the so-called atypical CML. So if we say that CML, chronic myeloid leukemia, is rare, 300 cases per year in Poland, this is 1 to 3 cases of those. So we have 1 to 3 patients per year in the whole Poland. And they are actually poor because they initially listen to the diagnosis, okay, you have chronic myeloid leukemia, so you will take 1 pill a day and you will be fine for many years, most probably. But then there comes a bad news. You don't have BCR-ABL. You also don't have JAK. So you have this atypical CML. And there are different, the so-called rare leukemias, but I will focus on this case because the question to us, can you confirm with next-generation sequencing that this patient has atypical CML, and maybe you can find something which we can use another drug of target. So we first look, of course, at the literature. We knew what to expect, and then we sequenced this patient. And what we found at first, we found TET2 mutation. So the most commonly mutated genes in atypical CML. And these are just few screens from the NGS. I don't think this is very impressive. You all have seen those integrated genomic viewer pictures probably many, many times. And we found also AS6L1 mutation within another usual suspect in a typical CML. We found SRSF2-1. And now the interesting story, because the doctor asked us, okay, so the patient has a typical CML, but does it have those mutations, any clinical relevance? And last year, few months before we got these results, the Teferis group from Mayo Clinic, they gathered a cohort of 25 patients. As I told you, this is a huge cohort in this type of atypical leukemia. And using just two simple things, and one not very simple, age, hemoglobin, and the presence or absence of TET2 mutation, they divided patient into so-called high risk and low risk. If the TET mutation is present, this patient is at a high risk. So what does it mean? The doctor should hurry up with the allogenic bone marrow transplantation. And actually this was performed in this patient. Fortunately, the patient had a sister who was compatible, who was in US, but who came. Unfortunately, I don't have the last, let's say, status of the patient, but I know he was transplanted. So just to sum up my talk, and especially in regard to the yesterday's discussion about the truth and the false, I would like to finish with this Edward Deming, famous American engineer motto. I would like to acknowledge my team, and also here is Tomasz Kurski team, but I don't know why it's not. It's probably the question of the screen. All the clinical hematologists, of course, the patients, and at the very end, I would like to show you one thing. On our immunology dashboard recently, one of the PhD students put an announcement that actually in the Polish supermarket, one of the cheapest supermarket, you can get more money than as a PhD student funded by National Science Center. So I agree, this is true, but definitely science is less money, but more fun. Thank you very much.
Tomek, thank you very much for this excellent lecture. Do you have any questions? Okay, here's one.
I was wondering if this was a phenomenon for chronic myeloid leukemia, that these stem cells are